department. I tried to group you guys in molecular. Not trying to leave sight out to the last here. I have to say, I don't know much about you, Mark, except that you mentor a lot of our grad students, undergrads, and we really appreciate it. And I know you work closely with the Bowman Lab. You probably have pretty fun lab meetings up at the end of them.
We do, yeah, thank you. And thank you for the introduction. So Mark Gomelski, I'm a professor in molecular biology. And as far as Justice Nelson goes, who's presenting next, imagine me receiving a call from a former student a few months ago who says that he enjoyed my class and he kind of wants to see whether there's any research opportunities. I did recall Justice, that he was a really good student, although this was a academic type course via Zoom. I still remember his pretty face, but more so, I did remember his on-target responses to my questions, which are often quite challenging questions. So he did have all the right answers. So I knew that he was a good sort of critical thinker. So I thought, yeah, sure, let's talk about it. And then I look at his student record, and he wasn't such a stellar student after all. So I thought maybe it was rushing too fast to offer him an opportunity. So I'm happy that it's proven that my hesitation was wrong. It's in the past few months, Justice has been working on a really fun research project that I think showed him that he has a passion for research. He's been taking classes, and if I look at his grades, it's all A's. So we have this transformation of a guy who seemed to have found his passion. I'm really happy when people find something that they love doing, because once you find that, there's no stopping, right? And so Justice and I, as well as Justice's mentor, here Trump, who's sitting right behind him, we're on a mission together with Zhang and Larissa on fighting cancer. So I'm happy to hear about Justice's progress in developing drones to fight cancer. Drones is just right up Justice's alley because he is also a lieutenant with the National Guard. So, Dan Pencil, Nelson, tell us about the drones.
Morning, everyone. I'm Justice Nelson. My project is about engineering Listeria monocytogenes into an anti-cancer drug. A little bit about me. I'm an Army officer in the Wyoming Army National Guard, as Dr. Mark mentioned. You'll see hints of that in the following slides. The very Army way to make points. I like being outside, but I did discover a passion for research and found out that when you do something you love every day, it doesn't feel like you're working at all. So, 50, 60-hour weeks feel like nothing, and it's really kind of a wonderful deal. So why do we choose to fight cancer? So cancer is the second largest killer of Americans, right behind heart disease, as indicated by the arrow. And the three frontline treatments we think about when we think of cancer are surgery, radiation therapy, and chemotherapy. These three traditional treatments come with shortcomings and side effects. Surgery, for example, can't reach all the metastases when a tumor begins to stage. Chemotherapy and radiation therapy often make the patient's quality of life sometimes unlivable. So immunotherapy is an emerging field, and immunotherapy seeks to treat cancer by taking the brakes off of the patient's immune system so that it can attack tumor cells and directly destroy them. Bacterial immunotherapy is an older field of immunotherapy, first noticed by William Coley in the early 20th century, who noticed that his cancer patients survived more often when they got a severe skin infection from the burn ward down the hall. So with bacteria, there are several candidates like E. coli, Salmonella, and Listeria, and bacteria like to live in tumors because it's protected from the patient immune system. And also because there's lots of cell debris, lots of things to eat, to use, to grow and reproduce. So we chose Listeria because Listeria is really unique. One of the unique things about Listeria is it is an intracellular parasite. So it moves very quickly into cells and takes up residence. You can see it on this fluorescent microscope image, Listeria inside the cell. And it also moves from cell to cell without exposing itself to the humoral immune system, right, from neutralizing antibodies. So you can see Listeria in green move on little comet tails of orange. So it can move through the cytoskeleton of the cell, bump up against the membrane, and then push through that membrane into the next cell. And then most surprisingly and most importantly in this project, Listeria accumulates selectively in tumors. So this is data from Dr. Gravecamp at the Albert Einstein College of Medicine in New York. And she found that Listeria hitches rides to tumors in immune cells. So when you inject Listeria into a body cavity or into the bloodstream, Listeria immediately moves into these immune cells, which then get recruited to tumors, and then from there, Listeria moves out to tumors. So more about these immune cells. They're called MDSCs, myeloid-derived suppressor cells. Tumors normally have MDSCs, it recruits them, and these MDSCs suppress the immune system at the tumor site. So they say, hey, there's nothing wrong here. Please don't come and send cells that are licensed to kill to come destroy us. So what happens is Listeria gets injected, moves quickly into these MDSCs, and it uses these MDSCs as an Uber straight to the tumor. And again, the Listeria break out from the MDSCs into the tumor, where it can use those little comet tails of actin to propel itself into neighboring cells. So what we're looking at is excellent penetration into the tumor beyond what we can do with things like nanoparticles. So I want to introduce briefly bacteria as drones, or Bactodrones. So the aim is get the bacteria to tumors. We're doing that with MDSCs as the Uber. The other important part of a drone is controlling time remotely. So controlling the drone in time involves using an inducer. In our case, it's a non-toxic antibiotic analog called anhydrotetracycline. And the other major part of a drone is multiple payloads. So just like a drone can do multiple jobs depending on what you strap underneath it, we're looking at strapping different proteins to Listeria to make it do different things for different cancers. In this case, the payload here is designed to alert the immune system and say, hey, there's something horribly wrong going on here. You need to send cytotoxic cells to the site to destroy tumor cells. So this brings me to my experimental roadmap for the spring and summer. So I started off bottom left with engineering the actual payload. That involves some modifications, some other things to get the protein to work in vivo and in vitro. Then I cloned it into a plasmid with an inducible system, so the backbone, same backbone for all of the payloads. Simplifies the process a little bit. I transformed that into E. coli and made sure our plasmid was built as intended with no mutations and no errors with colony PCR and sequencing, and then transformed it into Listeria and made the actual Bactodrone. And that's this big milestone right here. Following actually having the Bactodrone in hand, I took and did some experiments to see whether the drone functions as I intend remotely, and if it actually produces and secretes the protein. And then on to the mouse model, which is the big end goal up here. So the payload, in this case, it's a transcription factor called IRF3. So transcription factors are proteins whose only job in the mammalian cell is to be activated or modified in some way, move to the nucleus and bind to DNA where it induces the production of other proteins. So the reason we chose a transcription factor is because it amplifies what we can do with Listeria. So you absolutely can put a killer protein or a toxic protein or an immune signaling molecule directly into Listeria, but there's only 1 or 2 Listeria per cancer cell. So your ability to manufacture those proteins are really small. What we can do with a transcription factor is really amplify that signal. So we can put the full resources and machinery of the host cell, the mammalian cell, to work, producing those signaling molecules. In this case, with IRF3, it's type 1 interferon or interferon alpha. So the other part of the payload, you'll see on the next slide on the plasmid map, is control and time. So that's the inducible system. And then engineering IRF3. So IRF3 senses danger within the cell, normally in response to bacterial or viral infection. And as I mentioned before, it requires some modification to actually do that job. What I wanted to do with engineering this protein was turn the switch on so that it requires no modification, no processes in the cell. It just comes out of Listeria, goes to the nucleus, and initiates transcription. So the modified engineered form of IRF3 is called IRF3-5D. I synthesized that and cloned it into plasmid, PAJ in this case, by splitting the plasmid here, the promoter and the terminator, and cloning it in. Following the clone, transformed it into E. coli first, because Listeria is really not terribly fun to work with. So once I had transformants, I took a group of these and ran a calling PCR on it. Calling PCR just involves amplifying this portion of the plasmid with all the important parts in it. And that's about 2,100 base pairs. And you can see on this gel electrophoresis image indicated by the arrow, we have several colonies with the correct length of insert. But the length of the insert doesn't tell us about the orientation of the protein, doesn't tell us about mutations. So I sent off the plasmid for plasmid sequencing and came back and got 2 good colonies with no mutations, everything's in the right order. And those were colonies 1 and 2. So following having that good plasmid, transformed it into Listeria, and now I have the Bactodrone. So in order to see if the Bactodrone functions as I intended, I had to do some induction and secretion experiments. And here's the outline for that. So I took a colony, a culture of Bactodrones, and I split it into 2 parts. One part had no inducer, AHT. The other part had inducer, AHT. Incubated for 20 hours to allow the bacteria a chance to produce that protein, and then fractionated it. Fractionation involves centrifuging, and then you get a pellet down at the bottom of the tube and supernatant down at the top. The pellet represents all of the proteins and stuff that's inside the Listeria cells, and the supernatant contains everything that's secreted from the cell. So following that, there's a little bit of chemistry you need to do on the proteins before you can detect them on a Western blot. Ended up doing all the chemistry and putting them on a Western blot. The Western blot just separates proteins by mass and allows us to detect it with antibodies. So here's the result of that. So on the left, we see a really clear difference between no inducer and inducer. So this tells us that the drone is producing the protein we want, or a protein of identical mass, within the cell. If we look on the right-hand side of the slide, the extracellular fraction doesn't have much IRF3 with inducer. We do have this really cool degradation product, but that tells us that there's something going on with secretion. So one of the strategies we have on the Bactodrone team to improve secretion is adding a chaperone. And a chaperone protein is exactly like an adult chaperone at prom. It gets kids from one place to another to the right place in one piece, right? So chaperone proteins do exactly the same thing. They take our IRF3 propeptide and take it to the secretion system such that it can be exported from the cell. So on the left, we see still a clear difference, but we see more consistent expression. And we also see reduced degradation products. So that's a fun side effect of using a chaperone, is it also helps with polling and protects our propeptide from proteases getting cleaved off. Extracellularly, we also see an improvement in secretion. So again, this is a transcription factor. We don't necessarily need a ton of it. So there is some transcription factor here. Need to see if it works. And that's where we are right here. That's what I accomplished over the summer. Everything all the way up to secretion experiments right here. The thing left to do is the mouse model. So the mouse model is going to look a little bit like this. So we start with a mouse with a tumor. We inject the drones, and then we locate the drones. Locating the drones is proving to be a technical challenge because we're waiting on an imager, which is an instrument that can detect bioluminescent proteins within a mouse. It doesn't show up on the slide, but the tumor here is going green. So we're waiting on one of those imagers so we can stop sacrificing mice and grinding up their organs and looking for Listeria that way. So once the drones are where we think they should be, which is after 3 or 4 days, we then induce or activate the drones by putting the inducer in the mouse's food or water. Following that, there's measurements. So we measure not only tumor size, we also need to develop an assay to look at immune molecules. We need to see if interferon is actually being produced, if it's leading to the downstream cascade of effects and signaling that we're looking for with interferon type 1 signal. So in conclusion, Listeria monocytogenes is a promising anti-cancer agent that has the potential to change the way we think of frontline cancer treatment. Our drones are controllable. They go where we want by hitching rides on MDSCs, and they're controllable from outside the body, in vitro at least, with non-toxic chemicals. And now, the part left to see is whether the payload actually works, and then start making more. So I'd like to really thank the INBRE program and all the INBRE staff. It's been a really wonderful opportunity. Can't really express my appreciation enough. I'd also like to thank the Bactodrone team and the Gomelsky lab as a whole. A lot of people answering a lot of my questions. Terrific. And I'd be happy to take questions.
So, this is a transcription factor that's treating bacterial cells. So I guess I'm trying to understand, it's not a toxin, that's a direct. How is that going to get, is it supposed to get into it? How is that work exactly? Normally it would have to be internalized.
Right. Let me flip back a little bit. So Listeria is an intracellular pathogen, right? Yep. So Listeria gets into MDSCs, hitches a ride to tumors. From there, it infects cancer cells directly. So it lives inside those cancer cells without exposing itself outside. So then they produce these proteins. Wrong direction. Yeah, it produces IRF3 right here. Unfortunately, didn't get to talk a lot about IRF3 just for time and space. That's a great question. I do appreciate it. Cell culture models with Listeria are a little tricky because Listeria is not generally compatible with a lot of cell cultures. So there are things we can do like using BMDMs, bone marrow derived macrophages, but as far as a cancer model, there's not something good that we've been able to find and develop.
So Listeria, I do not know anything about this bacteria. How often does it go, if it had a choice, would it always go into the tumor cells, or would it occasionally go into other cells?
That's a good question. We found with mouse experiments over the summer, we're getting accumulation in most of the spleen, where we think bacteria are being destroyed by immune cells in the spleen. But yes, it does appear that they preferentially, almost always, the vast majority of the time, go to tumors and metastases. I think we have one more question. Yes.
Just to make sure, when you're injecting it into the mice, you're just injecting it really anywhere in the bloodstream when they're working their way to the tumor. You don't need to specifically inject it into the tumor, correct?
Yes, that's true. This is one of the really cool things about Listeria is you can just take and inject it intraperitoneally, and it doesn't even have to be in the bloodstream. It doesn't have to be directly inside the tumor, which is really cool because it allows us to get not only into the tumor, as you might also get with other direct injection methods, but also to metastases throughout the body, as far flung as the lungs, the mesentery, the liver, all kinds of places. Yes.
What kind of cancer tumor do you plan on using?
It's a good question. We're currently using 4T1, which is a mouse model of triple negative breast cancer. So it becomes really important because triple negative breast cancer, you may know, doesn't really express antigens that you can take advantage of to program the immune system to become destroyed. It's what's known as a cold tumor. So this is a really good candidate for the types of cancers we want to. Other questions? All right, thank you very much.